If you're a biology nerd or a plant, then you're in luck. Because today I'm going to tell you about my undergrad research project that has to do with Chlamydomonas single-celled photosynthesizers and how we trick them into producing oils. So what is going on? Let me explain. This is Chlamydomonas reinhardti. Chlamydomonas reinhardti. This. It's a single cell plant, basically. So it's not a plant. It's not. It's a single cell. And when you have large amounts of CO2, it's a happy Chlamydomonas, making photosynthesis and stuff. When it has low amounts of CO2, then it activates carbon capture mechanisms. So it's an unhappy Chlamydomonas trying to do whatever it can to continue producing sugar and survive. And it starts kicking off its carbon capture mechanism, CCM for short. So these are some molecular mechanisms for concentrating CO2 and still making some photosynthesis and surviving despite the stress. So it can kind of survive through a hard time. And then when you got really low CO2, then it almost dies and it starts producing oils. Later, when it recovers, when there's light, when there's CO2, then it will, start growing again and start consuming the oils and using it for energy. So there's a lot of interest through industry, because if you can make, say, a mutant that's always producing oils, then you can make really cheap biofuels and things like that without having to kill all of your cells, which is really cool because it could definitely help with energy production for cars and things like that. And this is why we're doing this project. Data analysis time! And this is where me and my group actually had a hand. So first off, let's talk about what the data looks like. So you have your gene, and we know that sequence, and then the result from the sequencing step is just going to be a bunch of little snippets all over with sequences that we can map out to the actual gene of it. And then we just count how many of those little snippets come from this gene compared to all the rest, all the snippets collected. So then we can have an idea of how important that gene is for the cell relative to all the others. Then we have our spells, our coding spells. This is why I like data analysis. Every single coding command is like a spell where if you do it right and you say it right and you wave your wand right, then it works and it's beautiful and it happens through mysterious forces that you don't want to learn about. If you don't know your spells, there's nothing you can do, and if you say your spell just a little wrong, it is not going to cooperate and everything is going to go wrong. So, the joys of coding. Anyhow, my favorite one is the PCA. So PCA is principal component analysis, and this is a statistical tool that kind of tells the machine to look for patterns in the data and organize and classify the data based on those patterns without actually showing you what those patterns are. And what matters here is that there are always patterns in data, it's just that they're usually so confusing and so weird that they're kind of impossible to report in any way that makes sense. So what they do is, without trying to explain it, they just show what they found with stats. And so it's kind of like machine intuition. And when you plot, we could see, like in dimension 1, so the main explanation of difference in conditions, so the different carbon levels and the different cell types. We had the mutant cell types all together and in a corner. And then we had all the wild type cells to the side and kind of spread out a little more. So they can tell that they're different cell types. And then also the low CO2 were all to the top and the high CO2 were all to the bottom and the middle CO2 were in the middle. So they also know that we treated the cells differently. That's so amazing. So this also confirms that their wet lab steps were all correct. Now we go into the Bayesian fit. The point of the Bayesian fit is telling whether or not something is different enough that you can consider it different. So for every gene that followed all these 3 conditions, we stored them in a list, and the list was 95 genes long. Yeah, so 95 genes here in the middle that fulfilled all of our conditions. I hope this was not too much information, or if it was, it's fun, it's cool stuff. I guess I'm a nerd. I hope this was a fun exposition to transcriptomics and genomics and biology research, but also how much power there is to bring stats and computation and engineering into biology research. And don't even get me started about all of the This is something I'm really passionate about. This is why I started a biology minor. I just did my course on this, and the prof had us do this and do all of this data analysis for the course, and I was hooked. And we talked to him. We thought it was awesome. And when we presented, we went over time and just kept showing more analysis that we did and like other fun stuff that we found with the data. And he was impressed, and he came to talk to us and said, all right, I think I have a project for you. This is why I think a lot of people come to UBC. It's to be involved in this phenomenal pulsing culture of research that's happening. Sometimes you won't be able to do research, sometimes the level of things is just too much, but maybe someone will have something for you. You'll never know until you ask. And so this is how we got started, and it's been really fun. So there are 3 of us that got approached. It was just our class group. It's been really fun. I really recommend, if you're into this kind of biotech field, to check out research, because biotech isn't all in the industry yet. Well, thanks for watching, and good luck with all of your happy and sad climbing emotises. Bye guys.